Good morning. It's January 30th, and this is your daily brief in science. Here's everything you need to know. A new study in Science argues genetics account for more than half of the variation in human lifespan, nearly double previous estimates, while still acknowledging a meaningful role for lifestyle and environment. Researchers developed a virtual twin framework to separate deaths from aging and external causes, addressing gaps in datasets lacking cause-of-death information. The study analyzes twin data to separate extrinsic mortality from intrinsic aging factors, isolating genetic influence on aging. A mathematical model adjusted for extrinsic mortality was validated with recent Swedish data, showing heritability increases as extrinsic mortality decreases. Denmark and Sweden twin data were used to calibrate how extrinsic mortality interacts with intrinsic aging. Experts describe the work as thorough and a meaningful extension of aging research from animals to humans, with independent scientists noting its rigor. The findings place the heritability of lifespan alongside other complex traits and suggest substantial genetic determinants of longevity. Healthy behaviors like regular exercise, a balanced diet, and strong social connections can modestly extend lifespan within a person's genetic limits, but cannot override an unfavorable genetic ceiling. Longevity is described as polygenic and complex, involving many genes rather than a single gene, with practical clinical applications decades away. Implications point toward identifying specific longevity genes and mechanisms governing internal clocks, potentially guiding therapies to slow aging and age-related diseases. Researchers highlight that substantial genetic influence supports large-scale efforts to map longevity-associated variants, refine polygenic risk scores, and guide drug discovery in aging pathways. A new study methodically tests whether adding Palbociclib to maintenance anti-HER2 and endocrine therapy can extend the time before the disease progresses in HR-positive, HER2-positive advanced breast cancer. The trial, named PATINA, reports a median progression-free survival of 44.3 months with Palbociclib versus 29.1 months without it, a statistically significant improvement. The NEJM publication titles the work Palbociclib for hormone receptor-positive, HER2-positive advanced breast cancer, and lists a multinational team led by Metzger and Mandrakar. PATINA is described as the first large randomized Phase 3 trial showing a clinical benefit of CDK4/6 inhibition in HR, plus HER2-positive advanced disease, suggesting a potential maintenance therapy approach for this subgroup. The trial results were initially presented at SABC's 2024, with the full protocol available on clinicaltrials.gov, NCT 029476685. A robust follow-up and a centralized database are highlighted as key supports for the mature progression-free survival analysis. Safety data show any grade adverse events in all patients, with higher grade 3 events driven mainly by hematologic toxicity. Dose reductions occurred in over half the participants, and serious adverse events were more frequent with Palbociclib, though there were no treatment-related deaths. These safety findings align with the known Palbociclib profile in this setting, and no new safety signals were identified. The reported hazard ratio for progression-free survival is 0.75 with a p-value of 0.02, indicating statistical significance. The study was Pfizer-funded and supported by an academic collaboration led by Alliance Foundation Trials, with Palbociclib not yet approved for HR plus HER2-positive ABC. Overall survival data are immature at the final progression-free survival analysis, with death counts similar between groups, around 60 versus 63, and a non-significant hazard ratio of 0.86. Leading clinicians say PATINA could signal a potential paradigm shift in treating HR plus HER2-positive metastatic breast cancer. A galaxy seen just 280 million years after the Big Bang has been confirmed by the James Webb Space Telescope with a redshift of 14.44, making it one of the earliest and most distant galaxies observed. MOMZ 14.4 is the most distant spectroscopically confirmed galaxy to date, placing it at Z equals 14.44, about 280 million years after the Big Bang. The discovery notes an exciting tension between observation and theory and points to new insights from a larger dataset of ancient galaxies. MOM-Z 14.4 is unusually nitrogen-rich relative to carbon, a signature that resembles globular clusters in the Milky Way and implies rapid early chemical evolution. Spectroscopy was crucial for precise distance measurements and for constraining the timeline of reionization, clarifying how early light transformed the universe's hydrogen fog. The study, Naidu et al. 2025. A cosmic miracle. A remarkably luminous galaxy at Z equals 14.44, confirmed with JWST, is cited, with Open Journal of Astrophysics publication noted in 2026. JWST has revealed more bright, ancient galaxies than expected, raising questions about early galaxy formation and the cosmic dawn. MOM-Z 14.4's brightness and chemical complexity, including nitrogen enrichment, suggest rapid chemical evolution and the possible presence of supermassive stars in the early universe. The discovery challenges existing models of early galaxy formation and star formation timelines, widening the gap between observations and theory. This finding extends the observable frontier and challenges theories of galaxy formation in the first 500 million years after the Big Bang. MOM-Z 14.4 fits into a broader trend of JWST expanding the observable universe and redefining expectations for the early cosmos. Follow-up observations led by Rohan Naidu at MIT confirmed MOM-Z 14.4's high redshift in nature. A NIH-funded expanded access program for ALS aims to provide MN-16 or ibutylast to patients ineligible for randomized trials while collecting real-world clinical data and biomarkers such as neurofilament levels. The SEANOBI program is supported by a $22 million NINDS grant under ACT for ALS and seeks to broaden access to MN-1-6, an orally available PDE4 inhibitor with anti-inflammatory effects. Medichinova reports that 100 patients have been enrolled across 12 U.S. sites, reaching half of the planned 200 enrollment, with ongoing challenges in recruitment and the usual uncertainties of clinical development and trial financing. The company notes that continued NIH support is critical to expanding access and advancing its development program, describing the SEANOBI milestone as meaningful progress in MN-1-6's clinical development and NIH/NINDS collaboration. SEANOBI runs in parallel with the COMBAT-ALS trial, a Phase 2b/3 study planning to enroll 234 patients in the U.S. and Canada, with top-line results expected by year-end 2026. The COMBAT-ALS design includes a 12-month double-blind period followed by a 6-month open-label extension, and Mayo Clinic references are noted in connection with ongoing ALS research collaborations. A new study pins down which animal lineage split first, revealing that comb jellies may sit at the base of the animal family tree. By comparing chromosome arrangements in sponges, comb jellies, and the closest single-cell relatives, researchers reconstruct the ancestral genome and track rearrangements since divergence. The key finding is that the comb jelly shows fewer chromosomal rearrangements relative to its inferred ancestor, suggesting it branched off earlier than the sponge. This places the comb jelly as the sister lineage to all other animals and identifies the earliest split in the animal lineage based on chromosome structure, rather than gene content alone. For decades, scientists debated whether sponges or comb jellies were the earliest diverging group. This study provides a chromosome-level perspective to settle that question. The approach focuses on how chromosomes are laid out, offering a different lens than purely comparing genes. The implications are significant for understanding animal evolution and the history of genome organization. By clarifying the root of the animal family tree, the work opens new avenues for studying the mechanisms that shaped early animal genomes and their rearrangements over time. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.